This is an exam question about inheritance and carrying out a genetic cross. It says two pure breeding strains of snapdragon, a garden plant, were obtained. One strain had red flowers and the other had white flowers. The two strains were crossed, yielding F1, so the first generation plants, all with pink flowers. The F1, the first generation plants, were then interbred to produce F2, the second generation plants, with the following petal colours. So we've got 62 red, 131 were pink, and 67 were white. It says the following hypothesis was proposed. Flower colour is controlled by a single gene with two codominant alleles. So if we just unpick this first of all, if we've got pure breeding strains, they are homozygous, meaning they only have one copy of the allele. So a red flower will only have the alleles for red, and a white flower will only have the alleles for white. These two flowers will not be heterozygous. F1 is the first generation that is crossed, and F2 is the second generation, and these are the results of the second generation. The question says, complete the genetic diagram to explain the cross. Use the following symbols to represent the alleles. So we're told that they're proposing that it's codominant. Now you represent codominant with a capital letter to represent the gene. So the gene in both these cases are petal colour, and superscript to represent the allele. And we've got two different alleles, an allele for red and an allele for white. Because these are codominant, if we had the heterozygous CR-CW, these flowers would be pink, because in a codominant cross, both alleles will be shown as a characteristic in the phenotype. Right, the question then says we've got to work out the parental genotype, the gametes, and then we've got to work out the F1 generation genotypes, the F1 generation phenotypes, and the gametes that they would produce. So phenotype is the characteristic that is shown, so that'll be the allele that's shown. So we've got red flowers and white flowers, so they're the characteristics that are visible. Genotype is what genes those flowers will be carrying. Well, we're told that these are pure breeding strains, so these are both homozygous. So we represent the alleles capital C small r, so this will be capital C small r, capital C small r. Every organism has two copies of each gene or allele. The white, again, is a pure breed, so that'll be capital C small w, capital C small w. And that's the parental genotype, what genes those parents in the F1 generation are carrying. Right, the gametes then, well, gametes are formed by meiosis. So you go from 2n in a cell to n, and they become haploid. So you halve the number of chromosomes, therefore half the number of genes. So if these cells divide into two, the only possible alleles that the cells can carry are CR. So all the gametes will be C, little r, C, little r. The white flowers, again, when you make the sex cells, the gametes, it's through meiosis, so the chromosomes are halved, therefore the alleles are halved. So again, the only options we've got is CW, CW. So there, our gametes, our sex cells. Right, the next stage then, we've got to work out the genotype of the F1 generation. So we've got to cross these gametes with these gametes. Now, you can see already that the red flowers are only going to pass on CR, the white flowers are only going to pass on CW, so all of them are going to be CR, CW for the genotypes of the F1. But just to show how we get that, we carry out a cross using a Punnett Square. So we represent the gametes of the red flowers across the top, gametes of the white flowers down the side, produce a Punnett Square, and if this gamete from the red flower joined with this gamete from the white flower, we'd end up with CR, CW. Same for this flower, this gamete from the red flower joined with this gamete from the white flower, CR, CW, and so on. So they're all CR, CW. So we have our genetic cross, and from that then we can write down our genotypes, what genes the offspring are carrying. Well, they're all CR, CW by looking at the Punnett Square. The phenotype is what characteristic will be displayed. Well, each flower has got a red allele and a white allele. We're told that they're co-dominant, so therefore when it's co-dominant, both will be displayed. So all the flowers in the F1 generation, the phenotype will be pink. Right, the next stage then is we've got to work out the F2 generation. So we're told, and we've worked out that the F1 generation is CRCW. So the gametes, again, the cells will be formed by meiosis. So it halves the number of chromosomes, therefore the alleles. So the sex cells will either carry CR or CW. So that's what alleles are found in the gametes. And we know we're crossing that with another flower from this generation because it says they're interbred. So CR, CW. So there are choices of gametes for each of the two parents. Again, we produce a Punnett Square, so we take the gamete alleles and place at the top of parent 1. Parent 2, we take the gamete alleles and put down the side. We create a Punnett Square and we work out what the offspring genotypes would be, what the possible offspring genotypes would be. So if this allele was joined with this allele, so these two gametes meet, then we're going to end up with CR, CR. Here we've got CW and CR, so CR, CW. Here we've got a CW and CR, so CR, CW. Can write this either way, but I'm just writing the R's first. And then the last one, CW, CW. So we've carried out our Punnett Square to work out the possible offspring genotypes. We're now asked to write down the genotypes. Well, we can see we've got CR, as one genotype of the offspring. So that's one done. We've got two that are CRCW. So we've got CRCW, CRCW. Let's put down them two. And the final one is CW, CW. So all we've done is copied our offspring from the Punnett Square and wrote the genotypes down here. We're now asked for the F2 generation to identify the phenotype, so which characters is going to be shown. Well, this plant has only got CR, and we know from up here that CR is red, so CR will be red. CRCW, the codominant, so those plants will be pink. Again, this is the same, so pink. And the final one is CWCW, so we know that is white. The final stage then, we've got to say what our expected phenotypic ratio is. So all we do is look at how many of these phenotypes we're going to actually have. So red flowers will have 1, pink flowers will have 2, and white flowers will have 1. And that's the phenotypic ratio for a codominant cross at the F2 generation. So that's this question answered then. We've completed the genetic diagram to explain this cross. Use the following symbols to represent the alleles. Alright, so we ended up with a 1 to 2 to 1 ratio, and the numbers they found during the experiment was 62 to 131 to 67, which is a very close ratio to what we found here. Therefore, we can probably say that this hypothesis is correct, but we would have to do some sort of statistical analysis like a chi-squared to confirm that this hypothesis is true.